Welcome to our deep dive into the fascinating world of blotting techniques. These techniques are like detective work inside a cell. They help us pinpoint specific molecules hiding within a cell's bustling metropolis. Today, we'll uncover how scientists detect specific biomolecules like DNA, RNA, and proteins using three powerful methods: southern, northern, and western blotting. Get ready to explore the world of gels, probes, and antibodies as we unlock the secrets of these techniques. Imagine being able to identify a single strand of DNA in a whole haystack of genetic material, or spotting a specific protein among thousands. That's the power of blotting techniques, and we're about to break them down for you. So strap on your thinking caps, science fans, and get ready for a thrilling ride through the microscopic world of blotting. First up, we have southern blotting, named after its brilliant inventor, Edwin Southern. Picture this. You want to find a specific piece of DNA, like a gene for, say, eye color, in a sample that's chock full of DNA. It's like finding a needle in a haystack, right? Well, southern blotting is here to the rescue. Our first step is to extract and cut the DNA into smaller fragments using special enzymes that act like molecular scissors. These fragments are then loaded onto a gel, kind of like a laboratory racing track. We apply an electrical current, and because DNA has a negative charge, it races towards the positive end. Smaller fragments zip through the gel faster, separating the DNA by size. Next, we need to transfer these separated DNA fragments onto a more durable membrane. Think of it like transferring a delicate fingerprint from a crime scene. This membrane, often made of nylon, acts like a sturdy piece of blotting paper. Now here comes the exciting part. We introduce a probe, a single-stranded DNA sequence that's complementary to our target gene. This probe is labeled with a radioactive or fluorescent tag so we can see it later. The probe roams around the membrane, and when it finds its perfect DNA match, it binds to it like two puzzle pieces coming together. Finally, we use X-ray film or special detectors to visualize the location of the bound probe. The result? A clear band on the film telling us exactly where our target DNA sequence is located. Southern blotting has revolutionized gene mapping, DNA fingerprinting, and diagnosis of genetic diseases. Now let's shift gears to northern blotting, named in analogy to southern blotting. Instead of DNA, northern blotting focuses on RNA, the molecular messenger that carries genetic information from DNA to build proteins. It's like eavesdropping on the conversation between DNA and the protein-making machinery. Similar to southern blotting, we start by extracting RNA from our sample. But remember, RNA is a bit more fragile than DNA, so we need to be extra careful. We then separate the RNA fragments by size using gel electrophoresis, just like before. The smaller RNA molecules dash ahead while the larger ones lag behind. Next, we transfer the separated RNA molecules onto a membrane, creating a mirror image of the gel pattern. Now it's time to introduce our trusty probe again. This time, the probe is designed to be complementary to our target RNA sequence, the message we want to intercept. The probe, labeled with a tag we can detect, searches for its RNA soulmate on the membrane. When it finds its match, it binds tightly, forming a detectable complex. This binding tells us that our target RNA is present in the sample. By analyzing the size and amount of the bound RNA, scientists can study gene expression levels, investigate the different forms of RNA, and even detect the presence of viral RNA in infected cells. It's like listening in on the cell's internal communication network. Last but not least, we have western blotting, the protein probing powerhouse. If southern and northern blotting are about finding the blueprints, western blotting is about identifying the actual construction workers, the proteins. Our journey begins by extracting proteins from our sample. We then use a technique called SDS page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Don't let the long name scare you. It's just a fancy way of saying we're using a special detergent to give our proteins a uniform negative charge and separate them based on size. Once the proteins are separated, we transfer them onto a membrane, just like in the other blotting techniques. But here's where things get really interesting. Instead of using a DNA probe, we use antibodies. Those remarkable proteins produced by our immune system to fight off invaders. We first introduce a primary antibody that specifically recognizes and binds to our target protein. Think of it like a lock and key. Only the right antibody will fit our target protein. However, this primary antibody is invisible to us, so we bring in a secondary antibody, which is coupled to an enzyme or fluorescent tag. This secondary antibody acts like a beacon, attaching to the primary antibody. When we add a specific solution, the enzyme on the secondary antibody sparks a reaction that produces a detectable signal. This signal, often a colored band on the membrane, tells us our target protein is present and even how much of it is there. Western blotting is a game changer in disease diagnosis, drug discovery, and understanding how cells function. And there you have it, the incredible techniques of southern, northern, and western blotting that revolutionize our understanding of biomolecules. From mapping genes to detecting proteins, these blotting techniques are essential tools in the arsenal of any molecular biologist. Who knew that something as simple as blotting could unlock so many secrets of the microscopic world? It's a testament to human ingenuity and our insatiable curiosity to explore the mysteries of life itself. Thanks for watching, and don't forget to like, share, and subscribe for more scientific explorations.